Hello students, today we'll discuss about second structure of prokaryotic cell, that is flagella. Now if we talk about flagella helps in movement of a cell. It has three components. One is this basal body which is embedded in cell membrane or cell wall. Right? Second is the hook which is the connection between the basal body and the filament part, and that is the third part of the flagella, that is filament. Right? So first is basal body. What is the function of basal body? Basal body is cylindrical structure which is embedded in the cell wall and the membrane. Second component is hook. Hook is the connection between the basal body and the filament, and filament is the longest structure of the flagella, which is made up of flagellin protein. That is the major difference if we talk about prokaryotic and eukaryotic flagella. Prokaryotic flagella is made up of flagellin protein, while eukaryotic flagella is made up of tubulin protein. That is the difference between these two flagella. Prokaryotic flagella is made up of flagellin protein, while eukaryotic flagella is made up of tubulin protein. Flagella is a feature, generally it is a feature of single cell. If the cell is motile, then flagella should be present. If a cell is non-motile, then flagella is absent. It is present in both prokaryotes as well as eukaryote. We have already discussed the difference between the flagella. Structure of prokaryotic and eukaryotic flagella is different. In prokaryotes, flagella is made up of flagellin protein, while in eukaryote, flagella is made up of tubulin protein. Next structure of these two cells are villi and fimbriae. Now, if we discuss the difference between villi and fimbriae and the similarity of villi, fimbriae, and flagella, all are membranous structure, right? Membranous extension. But villi and fimbriae has no role in motility. Motility means movement. It has no role in cell movement. If we consider movement of cell, then flagella is responsible for movement of cell, that is for movement. But villi and fimbriae have some specific function but other than movement. Both are smaller than flagella. Flagella is a longer structure as compared to villi and fimbriae. Now, what is the difference between villi and fimbriae? Villi is a longer structure, it is longer than the fimbriae, and it has major role in conjugation. Now, what is conjugation? Conjugation is, that is, suppose this is a first bacterium cell and it is second bacterial cell. This is the structure called plasmid. Transfer of plasmid from one bacterium to another through this tube. So this tube is termed as conjugation pili, and process of transfer of DNA from one bacterium to other, from one bacterium to another is conjugation. Right? So conjugation is process of transfer of genetic material or DNA from one bacterium to another, and pili is made up of pillin protein. Second structure is fimbriae. Fimbriae is, as compared to pili, it is a smaller structure, right? It is small bristle-like structure which is used for attachment to the host organism or maybe in rocks in water streams. So, in simple language, we can say function of pili is conjugation and function of fimbriae is attachment. Suppose that is the surface of rock and this bacterium wants to attach to this rock. So fimbriae helps in attachment to the rock and pili helps in conjugation. Next is membranous structure of prokaryotic cell. If we talk about membranous structure of prokaryotic cells, so first important membranous structure is mesosome. This is cell wall, and this second internal structure is plasma membrane. Extension of plasma membrane to increase the surface area. This structure is termed as mesosome, right? Mesosome is extension of cell membrane of prokaryote into the cytoplasm, towards the cytoplasm. This semi-fluid matrix is cytoplasm, right? So form of mesosome. Mesosomes are found in three structural form. One is vesicle, means spherical bodies. Next is branch tubule. Third is membranous lamella. These are three structural forms of mesosome. Now, most important for NEET, what are the function of mesosome? Now, what are the function of mesosome? First function of mesosome is mesosome increases the surface area. Why it increases the surface area of cell membrane? Because this mesosomal membrane has enzymatic activity for respiration. right? It has a respiratory enzyme or oxidative enzyme, and mesosome has a great role in respiration. For example, in eukaryotes, for aerobic respiration, there is an organelle, that is mitochondria is present, but in prokaryotes, since mitochondria is absent, mesosome has a major role for respiration. So we can say mesosome is analogous to mitochondria because of presence of respiratory enzyme. So first function of mesosome is it helps in respiration because of the presence of oxidative or respiratory enzyme. Second function is cell wall secretion, third is DNA replication, and fourth is cell division. How it helps in DNA replication or cell division? Suppose this is the genome of bacteria, right? This genome is associated with mesosome. This mesosome helps in DNA replication, and after replication, the DNA gets distributed to the daughter cell. So it helps in DNA replication and thus helps in cell division. So these are the function of mesosome. Next membranous structure is chromatophore. Again, this structure is cell wall and the internal structure is plasma membrane. Plasma membrane get extended to form this structure. These structures are chromatophore. Now, on this chromatophore, photosynthetic pigments are embedded like this. So basically, chromatophores are the site of photosynthesis. Photosynthetic pigments are embedded in chromatophore. That is very important. And it is present in autotrophic prokaryote, or we can say autotrophic prokaryote for photosynthesis. Generally, it is present in gram-negative bacteria. Due to presence of chromatophore, like there is one example we can write in which organism is it is present chromatophore. Example is cyanobacteria. So chromatophore, which is a membranous extension for photosynthesis, is present in cyanobacteria. Due to presence of this chromatophore in cyanobacteria, it is the connecting link between prokaryotes and photosynthetic eukaryote. Next structure is nucleoid. Nucleoid is basically nucleus-like structure. Nucleus is completely absent in prokaryotes, but instead of nucleus, they have a non-membranous structure. Means membrane is absent, but non-membranous centrally localized this genome is termed as nucleoid. This is nucleoid, right? So this, what are the components which are present in nucleoid? Nucleoid has DNA plus positively charged proteins. Why positively charged proteins are present? Because histone proteins are absent in prokaryotes. So for DNA packaging, positively charged proteins are present so that it can be present in a condensed form at the center of the cell. Present in cytoplasm of the cell, since nuclear membrane is absent, also known as, it is also known as genophore, or we can say incipient nucleus, or we also use a term that is primitive nucleus. So anything we can say, genophore, nucleoid, primitive nucleus, or incipient nucleus. In nucleoid, we have double-stranded DNA. This double-stranded DNA is, double-stranded means suppose this is the one strand and that is other strand of the DNA, right? Double-stranded and circular. Circular means two ends are connected. This type of circular DNA is present in prokaryotic cell and it is uncovered or naked. That means nuclear membrane is absent, right? There is one special note for NEET. Apart from genomic DNA means this nucleoid is genomic DNA. This is genomic DNA, right? Apart from this genomic DNA, prokaryotic cell have a special type of double-stranded circular DNA, and that double-stranded circular DNA is this plasmid, right? Now, what is the function of plasmid? Every DNA has some information for any type of phenotypic character. So this plasmid also provides special phenotypic character to the prokaryotic cell. It may or may not be present in a cell. Plasmid may or may not be present in a cell. If it is present, it provides special phenotypic character to the cell, right? And it is non-essential component of the prokaryotic cell. Why it is non-essential component? Suppose we have removed a plasmid from the prokaryotic cell, still the cell can survive. That is why it is non-essential component of the cell. That means it may or may not be present. It is extra chromosomal, it is double stranded, it is circular, and the most important point, it is self replicating. Self replicating means, suppose we initially have one plasmid, it can replicate and form two daughter plasmids, one strand of DNA and the other strand of DNA of the plasmid. So these are two daughter plasmid. So this property of forming the replica is replication. So we can say plasmid is self-replicating, extra chromosomal, double-stranded and circular. And this plasmid provides special phenotypic character to the bacterial cell. Example of special phenotypic character is antibiotic resistant property. Next structure of prokaryotic cell is ribosome. Now, ribosome are non-membranous structure. Non-membranous structure means any membrane is absent. Any membranous structure is absent in prokaryotic cell. Type of ribosome which is present in prokaryotic cell is 70S. Now, what is 70S or what is the structure of ribosome? Ribosome have this type of structure. This one is larger subunit and this structure is smaller subunit. Larger subunit is for 50S in prokaryotes and smaller subunit is 30S. Together, prokaryotic ribosome is considered as 70S ribosome. Now, what is the site of this ribosome? Where these ribosomes are present in prokaryotic cell? Site of ribosome in prokaryotic cell is cytoplasm and it is associated with cell membrane. Suppose this is cell wall and that is plasma membrane or cell membrane. So ribosomes are associated with these membranes, right? So it is associated with cell membrane, number one, and it is also present in cytoplasm. S in this is sedimentation coefficient. We'll discuss about sedimentation coefficient or Swedberg unit in the upcoming lectures. Now let us discuss about function of ribosome. Ribosome is mainly involved in protein synthesis. Function of ribosome is protein synthesis. That is why ribosomes are also known as protein factory of the cell. It is very important. Ribosomes are also known as protein factory of the cell. Ribosome also form a specialized structure or special structure in prokaryotic cell. That specialized or special structure in prokaryotic cell is polyribosome or polysome. This is a single mRNA. This structure is a single mRNA. Suppose that is a single mRNA and multiple ribosomes. These which I am drawing with red pen are multiple ribosome. Suppose multiple ribosomes are attached on single mRNA. That complex structure is termed as polysome, right? So basically polysome is nothing but multiple ribosome which is attached on single mRNA is considered as polysome. Function of ribosome is also protein synthesis. Function of polysome is also protein synthesis. Next structure is inclusion body. Now what are inclusion body? Inclusion body is present in prokaryotic cell. Instead of inclusion body, eukaryotic cell have microbodies. So that is the difference between prokaryotic cell and eukaryotic cell. Inclusion bodies are membraneless and microbodies are membrane bound. It is granular, insoluble in cytoplasm. Function of inclusion body is storage. We have four type of inclusion body which is present in prokaryotic cell. Number one is for storage of phosphate. Phosphate granule storage. Type of granule is volutin granule, right? If it is storing glycogen means type of carbohydrate. So it is glycogen granule. If it is storing Synophysin granule. Now, what is synophysin? Synophysin is a special type of protein, or we can say non-ribosomal protein, because ribosomes are not involved in synthesis of this protein. So, this is synophysin granule, right? Next is gas vacuole. Gas vacuole is also known as pseudo vacuole. Pseudo means false vacuole, which is present in prokaryotic cell. Why it is present in prokaryotic cell? It is present in aquatic prokaryotic cell or cyanobacteria. It provide floating ability. It is present in aquatic prokaryotes, right? So that is all about structure of prokaryotes. In next class, we'll be discussing about Gram staining procedure, right? Friends, today we'll discuss about Gram staining procedure. Gram staining procedure was given by H.C. Gram. And what is Gram staining? We'll discuss about the procedure and different type of bacteria on the basis of Gram staining. So on the basis of Gram staining, or we can say cell envelope and its thickness, on the basis of staining and on the basis of cell envelope and its thickness, there are two categories of bacteria. One is gram positive and the other is gram negative. So how we identify which bacteria or which category of bacteria is gram positive and gram negative? Basically, what is the procedure of gram staining? Suppose we have taken few colonies of bacteria and we are putting in the first step, we are putting crystal violet stain, or we can say the gram stain. The colonies will attain a purple color, right? In second step, we are putting iodine and 95% ethyl alcohol. After putting iodine and ethyl alcohol, few colonies will attain the color, that is the purple colony, and the other colonies will not attain the color. In third step, that is the second step of gram staining. In third step, we will put a stain, that is saffron, right? After putting saffron, these colony which loses the stain will appear as pink colored colony, right? And the other colony which was from the beginning were purple. So basically the colony which attain the gram stain and accommodate the gram stain even after washing with ethyl alcohol are gram positive bacteria. Basically, this ethyl alcohol wash off the stain, and gram negative bacteria, they do not accommodate the gram stain. So after washing with ethyl alcohol, they appear as transparent colony. After putting saffron, it take up the stain of saffron, not the gram stain. So the colony of bacteria which take up the gram stain are gram positive bacteria. The colonies which take up the gram stain are gram positive, and these are gram negative bacteria. Gram negative are the colonies or the bacteria which do not take up the gram stain. the gram stain are gram negative bacteria. Now, what are the major differences between gram positive and gram negative bacteria? Gram positive and gram negative bacteria. So, first basic difference is gram positive bacteria take up the gram stain and gram negative bacteria do not take up the gram stain. Right? Second difference between these two, in gram positive bacteria, cell wall is single layered, and this single layered cell wall is made up of peptidoglycan, while in gram negative bacteria cell wall is bilayered, means made up of two layers. It is made up of peptidoglycan and it is made up of thick peptidoglycan. In this, cell wall is bilayered with thin peptidoglycan layer. It is very important that in which category we have a thick peptidoglycan layer. A thick peptidoglycan layer is found in gram positive bacteria and a thin peptidoglycan layer is found in gram negative bacteria. In gram positive, mesosomes are prominent or most frequently found, while in gram negative, mesosome are less prominent. So these are the major differences between gram positive and gram negative bacteria. Next, in prokaryotic cell is shape of prokaryotic cell or shape of bacteria. If we consider shape of bacteria, we have 4 different categories on the basis of their shape. Number 1 is coccus or cocci. That means they have spherical shape. Like this. If there is single bacterium, then it is coccus. This type of 2 bacterium, it is diplococcus. Chain of bacterium, it is streptococcus, like streptococcus pneumonia. And if we have this group of spherical bacterium, then it is staphylococcus. Next shape of bacterium is bacillus, or plural is bacilli. If there is single bacterium of rod shape, it is bacillus. Multiple bacterium is bacilli. So shape is, in this shape is spherical, in bacillus shape will be root shaped. Now we have a special note about bacillus or bacilli bacteria. This group includes most of the bacteria. Most of the bacteria, right? Next category is spirillum, or we can say spirillum. Shape is spiral shaped, like this. Next category is Vibrio. In Vibrio, shape is coma shaped. The bacterium will look like this. Example of Vibrio is Vibrio cholerae. This is a single name, Vibrio cholerae. Vibrio is the genus and cholerae is specific epithet. Example of spirilla is Spirillum volitens. These examples are not that important. You need not to learn all the examples of the category. This is just the shape of the bacterium. If the bacterium is spherical, then the name is Coccus or cocci. If the bacterium is root shaped like this, then the bacterium is Bacillus or Bacilli. If the bacterium is spiral shaped, then it is Spirilla or Spirillum. And if the bacterium is coma shaped, then the name is Vibrio. One most famous example of Bacillus or root shaped bacteria is E. coli. E. coli is root shaped bacteria. And in spherical, Streptococcus pneumonia, Staphylococcus is the category of bacteria on which Alexander Fleming was working on. So these are the example of cocci. You can write Diplococcus pneumonia. Right? So these are the example of the bacteria. So that is all about prokaryotic cell. Now we will discuss about differences between prokaryotes and eukaryotes. Difference between prokaryotic cell or prokaryotic organism and eukaryotic cell. In first category we will write prokaryotic cell, and in the other category we have eukaryotic cell. Now what is the main or most important difference between prokaryotic and eukaryotic cell? Number one is the nucleus. Nucleus is absent in prokaryotic cell and it is present in eukaryotic cell. Instead of nucleus, prokaryotic cell have genophore, or we can say primitive nucleus. In this genophore or primitive nucleus, nuclear membrane is completely absent. That is the first and most important difference between prokaryotes and eukaryotes. We have discussed all the points in the structure of prokaryotic cell. We are just summarizing all the points of prokaryotic cell in the form of differences between prokaryotes and eukaryotes. Next difference is all the membrane bound organelles. All the membrane bound organelles are absent in prokaryotic cell. These membrane bound organelles are membrane bound organelles are present in eukaryotic cell. Next difference, for respiration, eukaryotic cell have mitochondria, right? Mitochondria is the site of aerobic respiration. Mitochondria is the site of aerobic respiration. It is site of aerobic respiration. But for respiration, prokaryotes have mesosome. So we can say mesosome is analogous to mitochondria. Next difference, for photosynthesis, prokaryotes have chromatophore. Photosynthetic pigments are embedded in chromatophore. For the similar process, in eukaryotes, pigments are embedded in thylakoid membrane. Thylakoid membrane of chloroplast, right? Next difference between prokaryotes and eukaryotes, plasmid provides special phenotypic character to the prokaryotic cell. This plasmid is absent in eukaryotic cell. Plasmid is absent. Right? Next point, or next difference between prokaryotes and eukaryote, or prokaryotic cell and eukaryotic cell. There is one special structure which is polyribosome are present in prokaryotic cell. This polyribosome is absent in eukaryotic cell. Next difference between prokaryotic cell and eukaryotic cell. In prokaryotic cell, cell division is through amitosis. Amitosis is the mode of cell division. Means there are three types of mitosis or three types of cell division: amitosis, mitosis and meiosis. We'll discuss about these cell division in the next chapter. And in eukaryotes, type of cell division is mitosis or meiosis. Mode of reproduction in prokaryotes is asexual, while mode of reproduction in eukaryotes, it can be sexual as well as asexual. Both can take place. It depends on the type of organism, which type of organism or which type of species it is, or the life, the phases of life. It depends on that. Next difference is gas vacuoles are present in aquatic prokaryotes. These gas vacuoles are absent in eukaryotes. Instead of gas vacuole, plant cell have true vacuole. That true vacuole is sap vacuole. These are the basic difference between prokaryotic cell or eukaryotic cell. Now, since we have discussed about all the points about prokaryotic cell, now we'll start with eukaryotic cell. An overview of eukaryotic cell. In eukaryotic cell, first we are going to discuss about a plant cell. Right? What are the component of plant cell? So first and outermost component of the plant cell is cell wall. If we remove the cell wall from the plant cell, the remaining component are protoplast. Right? Now, if we remove cell membrane from that cell, the remaining component will be protoplasm. Basically, protoplasm is nothing but nucleus and cytoplasm. Cytoplasm has two components. One is organelles and other is semi-fluid matrix in which organelles are embedded. And if we talk about nucleus has genetic material, basically DNA or chromosome stored in it, right? So this semi-fluid matrix, you know that it is the main arena of metabolic activity or biochemical reaction, right? Now we'll talk about organelles. What are the major organelles of a cell? So if we talk about organelle, we have three different categories. One is single membrane bound organelle. Second category is double membrane bound organelle, and the third category is non-membranous organelles, right? So in the first category we have endoplasmic reticulum, second example is Golgi bodies, third is vacuole, fourth is lysosome, fifth is microbodies. And there are some other organelles also, but these are the major single membrane bound organelle. If we talk about double membrane bound organelle, then first and most important is mitochondria. Second is plastid. What is the most important thing about plastid? It is present in plant cell and some of the protist, but not in the animal cell, and absent in animal cell, right? Next major difference, next major point about non-membranous organelle In non-membranous organelle, we have first example as ribosome, which is present in both prokaryotes and eukaryotes. But in prokaryote, type of ribosome is 70S, while in eukaryote, type of ribosome is 80S. We are talking about eukaryotic cell. So type of ribosome will be 80S in eukaryotic cell. We'll discuss about this. There are two subunits of 80S type of ribosome and structure of ribosome in the upcoming lectures. But first non-membranous organelle is ribosome. Next is centriole. Now what is the important point about centriole? We are discussing here about plant cell, but centriole is absent in plant cell. It is present in animal cell. Since we were discussing about all the component of cell, so I thought to mention about centriole also, that centriole is a non-membranous organelle, ribosome is a non-membranous organelle, and cytoskeletal elements. We'll discuss about all the organelles in this chapter in detail. Cytoskeletal elements are also non-membranous organelles. So that is the overview of a cell or eukaryotic cell. That outermost layer is cell wall. If we remove the cell wall, then the remaining component is protoplasm. Major difference between protoplast and protoplasm. If we remove the cell wall, the remaining component is protoplast. If we remove the cell membrane also, then the remaining component is protoplasm. So basically protoplasm is nothing but nucleus plus cytoplasm, and protoplast is the cell without the cell wall. So cytoplasm will be having organelles and the semifluid matrix in which organelles are embedded, and we have three different category of organelles: single membrane bound organelle, double membrane bound organelle, and non-membranous organelle. In single membrane bound organelle, we have endoplasmic reticulum, Golgi apparatus or Golgi bodies, vacuole, lysosome, and microbodies. In double membrane bound organelle, we have mitochondria and plastid. Plastid is present only in plant cell and in some protist. It is completely absent in animal cell. Plastid is basically for photosynthesis, right? Photosynthesis and storage of food. Non-membranous organelle, first category is ribosome. Type of ribosome is different because we are discussing about eukaryotic cells. So ribosome is 80S in eukaryotic cells. Second is centriole, and third is cytoskeletal elements. Since we are discussing about all the components, we'll quickly discuss about the difference between plant cell and animal cell. So first category is plant cell, and the second category is animal cell. It is very easy. You have been studying the difference between plant cell and animal cell from class 9th. First difference between plant cell and animal cell is cell wall is unique feature of plant cell, right? We have discussed it in cell theory also. Cell wall is present in plant cell and cell wall is absent in animal cell, right? That is the major and first important difference between plant cell and animal cell. Second, we have just discussed that centriole is present in animal cell, and this centriole is absent in plant cell. Next difference is plastid. Plastid is present in plant cell, or chloroplast we can say, and this plastid is absent in animal cell. That is for photosynthesis. We are heterotrophic organisms, so plastids are absent in animal cell. So since cell wall is absent in animal cell, hence outermost covering will be in animal cell will be plasma membrane, right? Next difference in plant cell and animal cell: number of mitochondria. Number of mitochondria are less in plant cell, since their energy requirement is less, the number of mitochondria are less, and in animal cell, number of mitochondria are more. Here it is less. Next difference between plant cell and animal cell, that is Golgi apparatus. Golgi apparatus in plant cell is called as dictyosomes, while in animal cell it is termed as Golgiosomes. Right? These are the basic main difference between plant cell and animal cell, which you at least should know before studying the structure of a cell or the component of a cell. Says that cell wall is absent in animal cell and it is present in plant cell. Centriole is absent in animal cell. Plastid is present only in plant cell, means plant cell and some of the protist. And number of mitochondria is less in plant cell because their energy requirement is lesser than animals. Golgi apparatus is known as dictyosome in plant cell and Golgiosome in animal cell. That is the basic major information about introduction of cell. Before we start about structure of a cell or component of a cell, in next lecture we'll be starting with first component of the cell, that is cell wall. Right? After cell wall we'll start with plasma membrane. After plasma membrane we will start with endomembrane system. There are 4 components of endomembrane system, that is endoplasmic reticulum, Golgi apparatus, lysosome and vacuole. So one by one we'll discuss all the component of a cell. Right?